So when we talk about the plasma membrane, we have to mention something called diffusion, and in a minute we're going to mention osmosis. And I think everybody's heard of diffusion, but we say it, we say things are diffusing, but how do we use diffusion in a biological context? Well, here it says diffusion is the tendency for molecules of any substance to spread out evenly into the available space. So really, I want you to underline of any substance on this sentence. And it's going to allow molecules of any substance to spread out evenly into an available space. So let's look at a picture to see what that's describing. So on the bottom it says, we have molecules of dye on the left-hand side of some membrane. Well, what happens is if you have a higher concentration of molecules on one side of a membrane than you do on another, because of diffusion, those molecules are naturally going to want to migrate to the other side. So as you move across this illustration, you can see that those molecules of dye are going to start moving back and forth until we get kind of a net gain where we get an equal amount of molecules on the left side of the membrane as we do on the right side. They're moving down their concentration gradient, if you will. And so when we're talking about the movement of any molecule down its concentration gradient across a membrane is what we're talking about right now, we're going to call that. So what's the difference between diffusion and osmosis? Well, osmosis is pretty much the same thing as diffusion, except for it's specifically talking about the movement of water. So here it says, osmosis is the movement of water across a semi-permeable membrane. Well, we're talking about the biological membrane, so it would make sense that when we're talking about osmosis today, we would talk about how water flows or moves across our cell membrane. And again, it says it can be affected by the concentration gradient of different dissolved substances that might be in our environment. So let's take a picture of that and see what they might mean by that. So here's kind of a crazy picture, and I'm going to break it down. But on top, we have, we'll say, a cup. And in the middle of that cup, we have a semi-permeable membrane. Well, on the left-hand side, we have little white and black dots scattered throughout our solution. And the white dots are sugar molecules, whereas the black dots are water molecules. Well, what you see is that there are more black dots on the right-hand side of the semi-permeable membrane than there are on the left-hand side. So, because of diffusion, or specifically if we're talking about water, osmosis, that is going to want to equalize. Those water molecules are going to want to equalize. So what happens is that these black dots, or these water molecules that are on our right-hand side, are going to want to move to the left across that membrane so that they can kind of equalize themselves. But what we have to consider is the fact that on the left-hand side of the membrane, we have sugar molecules. And for whatever reason, those sugar molecules are too big probably to move through our semi-permeable membrane. So they're going to stay on the left-hand side of our experiment here. So what happens is we get our water molecules moving to the left so that they can equalize, but our sugar molecules are too big to fit through our membrane. So we get kind of an unequal distribution of our aqueous solution here, or our water solution. And so if you did an experiment like this where you had solutes of, say, sugar on one side and then water on either side, you would see that at the end of the experiment, the volume would be higher on your left-hand side than it would be on the right-hand side. Now, when we talk about tonicity, it says water balance of cells without walls. Well, if we're talking about cells that don't have walls, we're talking about animal cells, right? Because plant cells are the only cells that have cell walls. So if you're an animal, you really have to be careful about how you treat your cells. And tonicity is the ability of a solution to cause a cell to gain or lose water. So we want to make sure that our cells aren't gaining too much water, and we want to make sure that they're not losing too much water either, because it has a great impact on the health of our overall being. So down here it says if a solution is isotonic, the concentration of solutes is the same as it is inside the cell. So what that means is that If I'm talking about a cell and I'm in an isotonic solution, that means that, let's say we're talking about salt. If I have 10 salt molecules on the inside of my cell and I'm in an isotonic solution, I'm going to have 10 salt molecules on the outside of my cell. They're going to be evenly distributed, and there's not going to be any net movement of water. Now, isotonic was our first example of how a cell can behave. There's two other examples we have to consider, and that's a hypertonic solution and a hypotonic solution. So let's break these down. Let's talk about a hypertonic solution first. Well, this is where the concentration of solutes is greater outside the cell than it is inside. So you might, it's not written on there, but you might put a little arrow behind greater and write outside. So the concentration of solutes is greater outside than it is inside the cell. And the cell is going to lose water. Well, that might not make sense, but let's try to break it down. So let's use our salt example again. It says we're going to have more salt on the outside of our cell. Well, let's say we have 2 salt molecules inside of our cell, and maybe we have 20 outside of the cell. Well, what happens, or what water wants to do, is it wants to diffuse those salt molecules. It wants those salt molecules to equalize. So the water inside of our cell is going to try to exit the cell to equalize the salt. And so it's going to push its way out of the cell, and it's going to say, hey, salt, we want you to get into the cell. It wants it wants to be in balance. But because of that, the cell is losing water. If water is exiting, our cells are becoming more and more dehydrated. So when we lose water, or our cells lose water, it's likely because they've been exposed to a hypertonic solution, or a solution where there's more solutes outside of the cell. Now, the exact opposite of that is a hypotonic solution. And it says here, this is when the concentration of solutes is less than it is inside the cell. So the concentration of solutes outside our cell is less than it is inside the cell. So, again, using our salt example, let's say we have 20 salt molecules inside of our cell and 2 outside. Well, again, water is naturally going to want to diffuse that salt. So it's going to rush into the cell, or towards that higher solute concentration. And what happens is that as water is rushing into the cell, those cells are going to burst, and that will destroy the cell. So that's a hypotonic solution. And there's going to be several illustrations to help kind of wrap your brain around that, because that can be pretty confusing. So here are the 3 examples we just talked about. Again, in an isotonic solution, which is the image on the left, you have an equal amount of solutes inside the cell as you do outside. So there's no net movement of water. Okay, water's moving in, but it's also moving out at an equal pace. In a hypertonic solution, however, we have more solutes outside of the cell than we do inside. So water's going to want to gravitate toward that higher concentration. And in the process, our cell is going to get kind of a shriveled appearance. In a hypotonic medium, we're going to have more solutes inside the cell than we do outside. And because of that, again, water wants to go to the higher concentration of solutes. So it's going to move into the cell, and eventually our cell could burst. Again, this is just kind of describing the situation we've been talking about. Now, how does water balance itself within cells that have walls? So we just talked about different examples in animal cells. Well, let's talk about plant cells now. How do plant cells respond to different kind of tonicity exposures, if you will? Well, cell walls actually help to maintain water balance, which is something animal cells don't have the ability to do. It says here, if a plant cell is turgid, okay, and turgid just kind of means kind of full, or, strong, if you're not strong, but just kind of inflated, if you will. If a plant cell is turgid, it's in a hypotonic environment. Okay, so remember, hypotonic was when there were more solutes inside the cell than there are outside. So it's very firm and it's a healthy state for most plants. So most plants actually prefer to be in a hypotonic environment. If a plant cell is flaccid or limp, it's in an isotonic or hypertonic environment. So here's an illustration of that. Again, it says if we have, this first picture is showing kind of a turgid or a normal cell. Okay, this is what we want our cells to look like. We want it to be in a hypotonic situation where water is continuously entering into the cell. We didn't want that in animal cells because our cells could burst, but in plant cells, because they have that tough cell wall kind of encircling the cell, we're just going to get a nice pressure, and water is only going to enter as it's needed because it's constantly going to be exerting a pressure on the inside of that cell wall. And that allows the plant to kind of maintain a vertical position. It allows it to reach up towards the sunlight, and it's just a really healthy state for plants. If we're in kind of an isotonic solution, which the second picture is illustrating, it's okay, but the plant could be doing better. Okay, water's entering, but it's also leaving. And then probably the worst environment a plant could be in is a hypertonic environment where water is continuously leaving the cell because there's more solutes on the outside than there are on the inside of the cell.